Hi, everyone, and welcome to the community webinar this week. If you're tuning into this webinar, this is the Healy Center's community webinar series. We host a couple webinars each month, and this week the topic is on the platform trial and specifically biomarker learnings. So the platform trial recently commemorated 5 years since its initial launch, and we've learned a lot in that time. So we have a couple really great guest speakers today who happen to also work at Mass General at the Healy Center. So once we have everyone in from the waiting room, the audience, I will introduce our guest speakers and we'll get going. And I think the numbers are starting to stabilize. So Mimi, if you wanted to head to the next slide. Again, thank you to those in the audience who are tuning in to our community Q&A webinar series. And we can head to the next slide. And the moment we are waiting for, so our two guest speakers today are Dr. James Berry and Eric Macklin. So Dr. Berry will start things off, and then Eric Macklin will come in with lots of interesting biostats information that goes beyond my understanding, but he'll do a good job breaking things down for everyone. I'm in the same boat. If you aren't biostatisticians in the audience, neither am I, but that's why it's a Q&A webinar. So Dr. Berry, I'll hand things over to you to get us started.
Thanks so much. Really excited to be here. Always a pleasure to be here and exciting to be here with Eric. Next slide. So just to remind everybody that the Healy protocol, the Healy platform trial has been going now for about 5 years. We've been really productive over that time. Each drug that we test is tested in what we call a regimen. So regimens A, B, C, D, E, F, and G each tested a different drug, tested against placebo, brought efficiency to that, efficiency to the startup, efficiency to the process, reduced number of people on placebo. So really the platform trial is doing what we hoped and expected that it could do. We also are sharing our successes with this through publications. So this is how we talk to other academics and bring advances like the platform trial, which honestly, was going on in oncology, and it was through some of those publications that we were able to kind of shape it to ALS, and we hope that the same will be true from, reading about it in ALS and taking it to new diseases and even creating potentially other platforms within the ALS community. So this is how we kind of spread innovation. Next slide. So as I said, we've had 7 regimens, A through G. Each of those regimens has been tested fully. Regimen C and D are considering phase 3 trials. The enrollment rate for each of these has been really far beyond what we see in most other trials, and that's because of the efficiency, because we have ready sites, because we can kind of hit the ground running. And actually, I was just having a conversation with an investigator from another disease area who just was, sort of shocked at the rate at which we were able to enroll and get answers for ALS. So I think it's just underlines really how we can think differently and do things differently. So that's an exciting thing about the platform trial. But there are more exciting things. Next slide. So while we're doing our job of testing these drugs and finding out which ones ought to go on for more testing and looking for effective treatments for ALS, at the same time we can learn about ALS and we can learn about how to make our trials better in the future. We can learn about the disease itself, and one of the ways we do that is by having what we call the integrated endpoint development engine, which is really kind of putting in novel endpoints, novel biomarkers, testing those, and then feeding that back into the platform trial and also other trials that are going on. So for example, we do whole genome sequencing. That genome sequencing will be shared with other researchers, adding to a growing repository of genomic data about ALS. Neurofilaments are done on all regimens, which tells us about the disease. We learn more and more about how to use neurofilaments all the time. We have other biomarkers that we can test in blood and spinal fluid. Some are specific to drugs, some tell us about ALS. And then speech analysis is an emerging digital biomarker or digital outcome measure. And what we've done here has really allowed us to work with a number of different companies using the platform trial data to try to understand what are the best ways to analyze that speech. And that's something that's going on now. And maybe we'll come to one of these webinars in the future and talk about the progress on that. Today, we're going to talk about another kind of digital portable mobile endpoint called home spirometry, which was really critical during the pandemic and allows us to look at how it works. This is spirometry means lung testing. This is when we have people blow into a tube and it tells us about how much air they can blow out and how quickly they can blow that out. We sometimes talk about the main parameter that we think about in ALS, which is vital capacity. And we've done some additional analysis really to tell us about this as an endpoint, particularly comparing home spirometry to clinic spirometry. And we'll have additional biomarkers and outcome measures for upcoming regimens. Next slide. So I think a lot of what drives our focus on digital health technologies like speech, like vital capacity at home, and like accelerometry and so many other digital health technologies is asking ourselves, what if we could use these digital devices to quantify people's functions and hasten ALS drug discovery so that we could do trials with reduced burden and increased accuracy? That's the what if. That's where it starts. And there are disease areas like cardiology where we can put on a cardiac monitor that's the size of a Band-Aid, maybe a little bit bigger, onto someone's chest and get data continuously for 2 weeks. And it's really transformed the way that we can think about cardiac monitoring and outcome measures for drugs that affect cardiac rhythms. And I think we can do the same thing in ALS, but we need to, there's some steps we need to do. Next slide. There is, an opportunity to analyze different data and analyze it differently in today's world where we have AI and we have ways of gathering data and contributing data using electronic devices, whether it's a cell phone or a tablet or a device itself. All of these can be internet connected and we can get really reams and reams of data and be able to analyze that data. We can also get additional scales, which we haven't talked about so much, but there are many additional scales that people could contribute without having to come to clinic to do that. Next slide. I think this just makes the point that we ought to take advantage of all of the data that we're creating and try to put that to use in clinical trials in a way that's logical. And when I say all the data, I think what we want to do is sort through all the data that we could collect and come up with the data that's most relevant. Next slide. So, these digital biomarkers might quantify function. They might turn our smallish data sets into big data. And then we can apply things like AI and large language models and machine learning to what becomes a very big data set from, what is an uncommon disease. We also want to simplify. Our goal is to go to simpler and simpler methodologies. And that's really how we do trials that are going to be less burdensome. Sometimes we have to start out with something that isn't quite as streamlined, but our goal is to get there. And these may facilitate doing trials faster, smaller, and they may eventually inform care as well. Next slide. So we know researchers, people with ALS, regulators would all like to move to digital outcome measures. And some are close to acceptance. Some of the things we're doing in speech are close to acceptance. Our goal, again, is to do these faster trials, less burden, and also reflect what's going on in real life. So we don't want to bring people into the clinic and do a test that, tests function, but maybe not any function that sort of impacts real life. We'd like to just sort of do it in the home environment if that's possible. But to be worth adopting, these digital health technologies or digital outcome measures have to improve on our current outcomes in some way. They have to be cheaper, faster, more accurate. And so there is a process of looking at how they compare to our current outcome measures. And there are some steps if we want to adopt these digital outcome measures. We have to gather data to show that it's useful, to show that it's better in some way. We have to feel comfortable with the usability. We might have a digital outcome measure that can give us good quantifiable data, but if it's not usable, it's not going to make its way into clinical trials. We have to understand not only what does the data show us, but what does that change over time look like? Because that's what we need for clinical trials. And then there are regulatory considerations that we have to keep in mind along the way. And with all of that in mind, next slide, we're going to talk about home spirometry. And I'm going to hand over to Eric to tell us about some of the details of what we're finding when we compare home spirometry, home vital capacity, to clinic vital capacity. In theory, home spirometry really helped us during the pandemic because we couldn't bring people in and do clinical measurements. It really mimics in-clinic spirometry almost exactly. It's a slightly different machine, but really the same measurement. And it can be done more frequently. But there are some drawbacks that we have to measure against that, which is that all spirometry, these are a couple of them, there may be others, all spirometry is subject to variability due to effort. So is it harder to standardize that at home? We know that coaching people in the clinic is really, really helpful to get the best effort. Can we do that well at home, or is that something that may be a problem? And the other thing is that small differences in technique can impact the results. We know that in the clinic, so we have people who are trained and expert in helping. And how does that translate when we do it at home? It was included in the HEALEY ALS Platform Trial due to necessity, but now we're analyzing it to learn about home spirometry itself and how can we incorporate this in future efforts. With that, I'm going to hand over to Eric.
Yeah, thanks. Next slide. Yeah, so that's a great intro, James. Thank you for setting that foundation. Just one step back, just to sort of frame it again. Why do we care about vital capacity? What is this effort that we're going through to try to see whether we can improve our assessment of vital capacity? Obviously, breathing is central to life. And unfortunately, the ability to breathe declines during the course of ALS. And we care about that because we want to know how we can best manage care for patients. And we also care because it might give us insights into which treatments, experimental treatments and trials are effective in slowing progression. So James already mentioned in a few of our regimens, particularly those that we were launching during COVID, regimens A through D, we implemented a program of collecting both in-clinic vital capacity when that was an opportunity, as well as using these home spirometers to collect data from essentially the same data, but from a different device and under a different context. And now we're in a position to try to see whether this more easily implemented at home spirometry, the force vital capacity at home using home spirometry, whether that performs as well as our gold standard in clinic slow vital capacity. Next slide. So just same as James already introduced, the spirometer is this little device that you blow into. It's connected by, I think, Bluetooth to a smartphone. And one of the nice features of the device that we chose for the platform trial is that it has a web interface or a smartphone video interface for the site coordinators to help coach that effort to try to maximize the degree to which there's equivalence to its use at home as there would have been had the participant been able to come into clinic. So there are 4 questions that the data we have, I hope we can use to answer, and they're focused on the degree to which home spirometry could serve as a substitute for our use of in-clinic vital capacity. Those are, do the 2 things, do in-clinic slow vital capacity and at-home force vital capacity, do they measure the same thing? If you have a measure of both, do they agree on what that vital capacity is? More operationally, in terms of a clinical trial, where most clinical trials set a threshold level of some minimal level of vital capacity to enroll a participant. If we were to substitute using home spirometry as opposed to in-clinic spirometry, would that give us would that identify the right people to enroll in that trial? Again, as James mentioned, as an outcome measure in these clinical trials and as a way to track the effectiveness of clinical care, we want to know how these 2 measures change over time. In principle, if they're measuring the same thing, they should, but there's different sources of variation in each. And as a statistician, I care about the data. What does the data actually show us about these measures? And then lastly, if we have 2 different measures, one is our home spirometry, one is our in-clinic, they're going to say something slightly different about a given participant, a given patient, given person living with ALS. Which one is telling us something more closely tied to their real underlying biology? Just your ability to breathe is not the whole, it's not the whole of you. And we want to find a measure that best tracks your underlying disease progression in a way that relates to something that we're ultimately trying to control, survival. Okay, next slide. So this was our data we had out of these first 4 regimens. We looked at all the participants who had matched in-clinic vital capacity measurements, slow vital capacity, and at-home assessments that were within 14 days of each other. We restricted ourselves to people who had at least 3 or 4 of these matched assessments so that we could look at the change of these measures over time. And we only used those assessments for home vital capacity that were adjudicated and reviewed and judged acceptable by a central reader at the BNI Outcome Center. So not only were these, not only was it this FDA approved device with coaching by a trained site coordinator, but also there was after the fact an evaluation of each of the flow, what is called a flow loop, your ability to produce a breath by central readers who are trained in this. Out of that, we got 166 participants with those characteristics, and they tended to be relatively early in the disease course, only less than 2 years from symptom onset. They tend to be a little bit slower than a typical clinical trial participant in rates of progression or prior to baseline, because we were only focused on those who had completed all follow-up. And as you see at the bottom of that table, folks on average had lost about 20% of their lung function relative to someone not with ALS. Next slide. So this is the first question we're looking at. Do these two measurements agree with each other? So this is just scatter plot on the right. On the x-axis is the measured vital capacity in the clinic. On the y-axis, the forced vital capacity measured at home. And that diagonal dashed line is the one-to-one. So if every point lay right along that line, that would be perfect agreement. Obviously, there's some noise. The correlation, which is a measure of how well they agree with each other, is 0.8, ranges from -1 to 1. So that's a pretty good correlation. There's no systematic bias. They're almost right on top of each other on average. There's a little bit more variability in the forced vital capacity measured at home than in clinic. There's a little bit more people at the lower end of the FVC and the high end of the FVC than there are. But they, cross-sectionally, if you just compared these two measures, they're working very well. So that's a check there. Next slide is our next question. Next slide, please. So then making this operational in the context of using it as the eligibility criterion for a trial. For the platform trial, we used a threshold of 50% predicted forced vital capacity. Because we excluded everyone who was below 50, we don't really have the data to evaluate at that point. So this little figure is looking at what if we had some hypothetical trial where a 60% predicted was the threshold for eligibility in that trial, which is relatively common in other ALS trials. And along the diagonal are the places where those two measurements agree. In the upper right and the lower left, the 73% and 14%, those are places where both measurements say this person should be eligible or this person is not eligible. And if you look across the top row, those are the folks who by home spirometry would have been judged eligible. And what percentage of those? 73 out of the full 78 is 90% or more. So this home spirometry is working very well in identifying people who should get into the trial. The last piece, though, didn't perform quite as well. It's looking at the bottom half of that graph and it's saying of those for whom home spirometry said they weren't eligible, were they in fact not eligible? And you'll see that there's a relatively large fraction, that 8% relative to the 14%, of folks who would have likely been eligible if they had gone into clinic and had a test. Now, these are all people who did go in the clinic. So I'm sort of applying these to the thought process of how could this perform in the future in some other context where we only had at home spirometry. We might lose some people from those trials who could have qualified if they had gone into the clinic. So it's not a perfect measure on this score. Mostly we're losing people on the bottom end. Next slide. The next question is, perhaps more important is, do they track the change in how patients are functioning over time? And the correlation, this is again, the same sort of can you back one before we get to the orange and green? This is another scatter plot. Now we're looking at slope. So people an average change over time. Are you trending up? Are you trending down? Are you trending down quickly or slowly? And so this is in percent predicted per month. It's a little bit below zero. People, unfortunately, are progressing slowly over the course of our observation during their participation in the trial. And the two measurements agree not quite as well. So again, this correlation measure can range from -1 to 1. Here the correlation is 0.5. That's not bad, but it's not maybe as good as we like. You can see more scatter around the 1-to-1 line. And not only is there not sort of perfect correlation, but there's a little bit of a bias where the estimate of the rate of change by home spirometry is about 30% slower. Now, is that good or is that bad? We don't, what's the truth? What we have is what we're familiar with is our in-clinic slow vital capacity, but we don't just from these data know which is telling us the right thing, which is giving us the right information, the information that's most insightful about that patient's disease progression. And so then if you do click to those colored dots on this slide. So I want to pick out as two people as a contrast. So there's one person in orange who's if they were measured in clinic, you would say that they were progressing by about 2 percentage points per month by in-clinic spirometry, but by about 4 or 5 percentage points if you were tracking them by their home spirometry. And the person in green is the converse. So they're progressing by about 4, a little bit more points per month in clinic, but they're only progressing about 2 points per month by their home spirometry, their forced vital capacity at home. And the question is, which of those is a better predictor of how long that individual survived? And we'd like to believe that if we're measuring something that's really close, really insightful about their disease process, that should be a good predictor of what their survival time is. Next slide. Next slide. Yeah, thank you. And so there's not a nice graphic for this table from someone involved analysis that then says, let's look at these slopes from the two different measurements and see whether one or the other predicts the time to survival, the survival time of each of those participants, better or worse. Each of them on their own explains about 10%. So the rate of change over 24 weeks, the average rate of change over 24 weeks in either in clinic or at home spirometry explains somewhere between 9 and 10% of the variation in survival times. So a little bit worse, only 9% for at home, 10% versus in clinic. If we adjust for other things that we know about these participants, like how long they've had the disease, how quickly they were progressing before we started following them in the trial, whether they had bulbar onset or an impairment of the ability to speak or swallow. So there's a range of features that are predictive of someone's survival that's subsumed within this measure called the tricals. And if we adjust for this other known set of predictors, then forced vital capacity measured at home only explains about an additional 5% of the variation in survival times, which is only about 60% as good as what we would have learned had we used slow vital capacity. So again, not quite as good performance, but that's going to be balanced by other features. So next slide sort of wraps things up. So what do we conclude? Slow vital capacity probably does deserve to remain our gold standard at this point from what we've seen here. FVC is not a perfect substitute for that. There are things that we would have missed or misjudged if we'd relied solely on home spirometry and at-home FVC. But home spirometry is much more convenient, and it reduces the burden on participants substantially if they don't need to come into clinic. And it allows us to reach many more individuals out at their homes than are able to come into one of our clinical trials at a big tertiary care center. So there's a lot of benefit operationally, and maybe we can compensate for some of the inefficiency from home spirometry by increased numbers of participants who we could get involved. And it's that understanding of the trade-offs between these two measures that I think will advance our efficiency in developing trials going forward, so that we can do the best job, most efficient job of trying to answer the questions that will lead to better treatments and ultimately, we hope, a cure.
That's great. I think that's the last slide, right,
That's
Eric?
the last slide, yeah.
That was terrific. Let me ask one other question, and then we have a bunch of things in the Q&A that we can get to. But one of the things that we often talk about is how frequently we get a measurement and what's the impact. Let's say, for example, that we think it maybe is a little more variable to measure at home, but what if we could do that 3 times as frequently as in clinic? Is there a benefit there or no?
Yeah, there's probably a little benefit. So we don't have those data from this study. I know you and I have worked on other studies where we do have much more frequent assessments, and we can ask explicitly from the data the degree to which we can improve precision by having frequent measurements. It's not very much. It's not as much as one would wish, because most of the variation is person to person. One person's disease course is different than another, and regardless of how many times you measure it, that's still the case. The person who's progressing slowly is going to progress slowly, and we'll have a relatively precise measure of that. And the person who's progressing more quickly, unfortunately, they're progressing more quickly. And just having more density of data, more data points to see that doesn't reduce that source of noise in the system that makes it more, that leaves us still needing a bunch of participants in a trial to answer whether treatment was effective on average among them.
All right, so I want to get to a couple of the questions in the Q&A, and some of them are sort of more for me, but we'll go back and forth. So there are a couple of questions that are about the comparison of SVC versus FVC. So we did SVC in the clinic and we did FVC at home, and I guess I would just clarify from a clinical perspective, they're very, very close to interchangeable. There are some studies that show that they correlate very, very, very closely. There are theoretical differences. So FVC is forced vital capacity and SVC is slow vital capacity. And the difference is just in the procedure. So forced vital capacity, you blow out as fast as you can. And when the machine detects lower than a certain amount of flow of air, it kind of cuts off and says, look, this person's done. Slow vital capacity, you blow at a more steady rate and you don't have that cutoff for the tail. And so if someone has spasticity, then theoretically there's a difference. In practice, it doesn't seem to be as big a difference as one might think. So they're pretty close. It does leave open, what we like to do in science is test one variable. And so it would be very nice to test the one variable of slow vital capacity at home or slow vital capacity in the clinic. And I think it's an astute question. But the reality is that the machine just didn't have a setting for SVC. So I feel pretty confident that they are very, very close to the same thing, but that's the complete answer. Eric, I don't know if you have anything to add to that.
No, that's no, nothing to add on that one. No.
So there's one person who just noted that their breathing has been, very, at a low but stable level for a long time, and that they've been excluded from projects because of this. But yet there are people who have a higher number at the outset and then have more of a decline. And this I think highlights one of the things that is a statistical challenge for us. It's a real challenge for people trying to get into trials. It's a statistical and a design challenge for us to try to include as many people as possible and yet try to get some sense of homogeneity or similarness between people who are entering. And the reason we do that is to try to boost our signal to noise ratio, the noise being differences between people and signal being, is there a slowing effect of a drug? But it hits on something that's really challenging for us from a humanistic perspective, from a statistical perspective, and from a trial design perspective. Eric, do you have any comments there?
Yeah, I can just echo what you're saying, that I think our overriding goal is efficiency. We'd like to get an answer as quickly as we possibly can. These things, as you probably know, if you're on the webinar, you're tracking it closely enough to know that these are still very slow processes. The time, as efficient as the platform trial is, We've studied 7 different compounds. We haven't identified one that we think is really remarkably effective. That's great efficiency, but it's still 7 years, 5 years, I guess, coming into our 6th year. That seems like a long time when you have ALS. So a lot of our trial design design thoughts are around how can we do this as fast as possible. And so just, again, sort of expanding on what James has said, imagine we wanted to know whether an exercise program could increase, could reduce your 1 mile race time. If we enrolled patients who were ages 3 to 93, it would be really hard to see whether exercise was going to have much effect, because some of those people in that group are not going to take to the exercise very well. If you're 3 years old, you're probably not listening to the coach. If you're 93, you might be listening, but you're not actually getting much faster. But if we just took folks who were, 20 to 25, just out of college, we might be able to detect a very strong effect of that exercise program among that relatively homogeneous group. And so that's the same sort of principle. It's disadvantageous to people who don't get in the trial if they were hoping to, but it allows us to move the field forward as quickly as we think we can and still address the broad question of, is this therapy beneficial for ALS patients generally?
And just to expand a little bit on your analogy, we've done a lot of modeling for platform trial, a lot of kind of looking at the data so that we can expand beyond 20 to 25 and say, well, actually, if you go from 18 to 42, there's actually no difference. That's fine. You can enroll all of those, when we might imagine that, well, it really has to be people in their early 20s. So we've been able to actually expand some of our parameters by really looking at the data and being thoughtful about, what benefit do we get by restricting things, and really trying to loosen it where we can. So that's kind of how we've tried to go about this. There are a couple other questions, and then we're going to have to close for time. But one is about a path to deploy home spirometry as an official metric in trials. I think there are really a couple of aspects to this. One is doing the work that Eric is explaining now, which is a work in progress, which is to say, how does it perform? And then really do some thinking scientifically about, is the performance versus the kind of the downsides versus the operational benefits worth it? And that might be different in different trials. It depends on the context. There is a regulatory component. So what do regulators think about it? And that happens in conversations as we design it into trials and have those conversations. And it's really a second step after we say, yeah, we do think that scientifically we should put it in. There's an operational step to say, what do we have to do to deploy it? And that often gets easier, actually. I have to say that sometimes these products get easier to use, less confusing, and there may be a pathway. So I think those 3 things have to come together. But the first step is this scientific exploration of what are the scientific trade-offs. Eric, did we notice any differences between bulbar onset patients and limb onset patients, or have you looked yet? It might be just a good idea that we ought to look at.
The patients with bulbar onset progress more quickly, and so we can see that in both measures. I have not done a thorough analysis of whether the degree of agreement in forced vital capacity at home versus in clinic slow vital capacity is better or worse in bulbar or more likely in limb onset patients than in bulbar onset patients. We're also, as you've smartly suggested, not only is the site of onset may be relevant, but what was the participant's function at the time that we started tracking them? Someone who has limb onset, by the time we see them, they already have some substantial bulbar dysfunction. That might be more relevant to us than the actual original site of onset. So there are a number of other features of the participants that might help us resolve whether there's a subset where forced vital capacity works really well and another subset where it doesn't work so well, distinct from this general presentation I gave today.
And a hypothesis might be that if people have bulbar involvement, they may have a hard time making a seal, and we might get more variability. We may manage that better in clinic than we do at home. But the really amazing thing about the platform trial is that that's a hypothesis, and we have the data so that we can test that. And we prove ourselves wrong all the time. And that's as exciting as proving ourselves right, I think, to a researcher, because it gives you a path forward either way. And so that'll be really, I think, very, very interesting to see. It's an astute question. The last one, I just want to reach out about one that was a little bit unrelated, but about the whole genome sequencing. What does it cover? Does it cover, the main genetic mutations or really rare ALS mutations? So that whole genome sequencing is done, and we store it for the trial. We sort of get all the routine mutations called. We call it calling it. So they basically comb through that genetic code and tell us whether the common and known genetic mutations are there. And that's data that we've already looked at. Then, to make better use and more use of the whole genome sequencing, that whole genome sequencing is put together with other whole genome sequences of people with and without ALS, and we can do exploratory work to look for new genes, to look for these very, very rare changes, and under the right circumstances, potentially look for things like a genetic response pattern to a certain to a certain drug. Those are a little complicated. I don't want to make it sound easy, but there's a lot we can do, and so gathering it is really, really an important thing. All right. Final thoughts, Eric?
Oh, excellent. I appreciate everyone's involvement, correspondence with us today. James get to see patients in clinic. I don't get to see many people, so it's always a pleasure to have the chance to connect with you guys. So we're all in this together.
Absolutely. And Eric, it's such a pleasure to work with you. I mean, you know the disease so well, and it's been a long partnership to sort of really try to understand things better and try to design better trials. There are more questions. We can't get to all of them. I'm sorry, but we'll try to follow up. So thank you to everybody for being here.
I'll chime in quickly, James, and just say for those who have enjoyed delving into the data from the platform trial today, we've got more of this coming. So September 25th, we'll have another platform trial webinar. We're going to have patient advisors and research ambassadors do a panel discussion about the significance of what placebo data retirement means for the platform trial and what that data sharing does for the field of ALS research. So that will be a great one in September. The following one, I think it's October 23rd, we're going to have Dr. Lori Chibnik, who works closely with Eric Macklin and Biostats, and she's going to go through how do you interpret these research articles that we've been publishing about the trial. So there's all the results from those regimens that Dr. Berry was talking about. They've been published in scientific journals, and how do you make heads and tails of that? So that one in October will also be a great way to kind of dig into what we've learned so far. But with that, thank you again, Eric, and thank you, James, for being here. And thank you to everyone who attended and is being part of this research conversation and helping push the field forward.
Appreciate it. Thank you.
Thank you.